Hi, everyone. I'm Nancy Quarterman, the founder of the Usher Syndrome Society. I have 2 children who are living with Usher syndrome, type 2. The Usher syndrome raises awareness, public awareness for Usher syndrome through the portraits you see. We have a global exhibit, we do film, we do educational events, and we also raise funds for research. And this year, we committed about $500,000 to support translational research grants. And I am pleased to announce next Dr. Jeffrey Holt, who I've worked with for many, many years. He is a professor and director of research in the Department of Otolaryngology at Boston Children's Hospital and Harvard Medical School. He is the chair of our Usher Syndrome Society Scientific Advisory Board committee, and he, for our Usher Syndrome Society Translational Grants. He will be giving updates on the 3 current projects that we are working on. So here's Jeff.
Thank you, Nancy, for the introduction, and thank you all for your attention this morning. It's a real pleasure for me to be here and to be able to share some of these updates. So, in my role as chair of the Usher Syndrome Society Scientific Advisory Committee, I would like to tell you about what we've been up to. The board of the Usher Syndrome Society are pictured here, and it's been really an honor for me to be able to work with this group to start thinking about how we can develop new therapeutics for treating Usher syndrome. And so we began this maybe 1.5 years ago and started discussing the possibility of funding translational research. And so through those discussions, we formed the Scientific Advisory Committee, which are pictured here. I am the chair of that committee. Shannon Boy, you just heard from, is one of the members. Ben Skyorki is in Basel, Switzerland, and he studies retinal disorders. Elliot Shearer is one of the clinician scientists at Boston Children's who helps us with this work. And Teresa Nicholson is at Stanford University and studies zebrafish. So the initiatives, some of which are listed here, appear on the Usher Syndrome Society webpage. And so I'm going to focus on this one with the colorful looking spiral. That's a cochlea, and you can see hair cells in green and neurons in red. This is one of the images from my lab. But let me tell you about the funded research and the plans. So we generated what's called a request for applications, which was circulated throughout the scientific community. That was July last year. And the goal was to fund translational research to develop treatments for all forms of Usher syndrome. As Nancy mentioned, there was $500,000 that was dedicated to this program. And so we got 26 letters of intent from scientists around the world, and we narrowed it down to 11 that we invited to submit a full proposal. So the Scientific Advisory Committee reviewed those proposals, and there were many good proposals there, but we selected 3 for funding. And so that funding began in January of this year, and so we're just 6 months into these projects. So it's early days, but I would like to tell you a little bit about each of the projects. So the first project is, the title of the project is A Screen for Compounds That Prevent or Slow Retinal, Auditory, and Vestibular Cell Death in Usher Syndrome. And this is being done at the Institute for Neuroscience at the University of Oregon, and it's headed up by Monte Westerfield and his colleague Jen Phillips, and they have 2 students who are working on this, Eric Fox and Sarah Buckner, who just recently graduated. And so their idea is that genetic defects in Usher syndrome proteins lead to stress within the cell, and cell stress can eventually lead to death of the cells in the ear and in the retina. And so their goal was to identify drugs that are already approved by the FDA, the Food and Drug Administration, that can protect against cell stress and prevent cell death. So by addressing this, they may be able to identify drugs already approved, and they may be effective against multiple forms of Usher syndrome. And so the second goal was to test these drugs that have been identified for their ability to prevent cell stress and death of hair cells and photoreceptors. And so they're using the zebrafish. You've heard about the zebrafish a little bit, but the zebrafish, I think, is actually a very good model because the sensory hair cells in the ears of the zebrafish and the retinal photoreceptors are actually quite similar to those of humans, and the genetics is also quite similar. The zebrafish Usher proteins are all expressed just like they are in humans, and many of the mutations generated in a zebrafish cause the same set of symptoms. The other reason for using zebrafish is they're very inexpensive. So scientists can screen thousands of fish, and that's much easier to do that with a small animal that you can do much more quickly. And so for the project, several medicines have been identified, and one that was found that improves visual function. And so here are the data, just quickly to show you this. On the left, you can see normal visual function, and the fish carrying an Usher1F, in this case, mutation, shows reduced visual function. When the drug is applied in the normal condition, showing that there's safety or no change in function, that's the third column. And on the far right, the Usher fish with the drug show improved retinal function. So this was an encouraging result. This group also looked at the retina, which you can see right here, and looked at the photoreceptors to see if there was preserved numbers of cells. And so they counted the cells, and they didn't see a huge difference in this case. But the fact that the previous data set showed that there was an improvement in retinal function was encouraging. And so their preliminary conclusion, just 6 months into the project, is that reducing cell stress can improve visual function in this zebrafish model of Usher syndrome. And so the next steps are to identify other medicines, other FDA-approved drugs that can promote cell survival. They also want to evaluate medicines for additional forms of Usher syndrome. They've tested it thus far just on Ush1F, but again, their hypothesis is that by targeting this cell death pathway, maybe they can prevent cell death for multiple forms. And then eventually they want to evaluate this for hearing and balance in the treated fish as well. So the next project is called Rescuing a Common Deep Intronic Usher2A Variant by Enhanced Deletion Cas9. This is a CRISPR form of genome editing. And so this is being done in Germany at the University of Tübingen, and it's being done by Susanne Kohl and her colleague Pietro de Angeli, as well as a student, Stefania. And so their idea is that a new form of gene editing can repair an Usher2A mutation. This is the name of the mutation here, and they're hoping that by editing that gene, they can remove the mutation and lead to healthy RNA production and healthy Ush2A protein formation. This particular mutation accounts for about 4% of all Usher syndrome mutations, so it's actually fairly common. And so this is a little bit complicated slide, but let me just walk you through this. You've heard about exons, and so exons are portions of DNA that actually code for the protein. And when that protein is being made, the exons get spliced together to form a full-length proton. Now, this particular mutation occurs in between exons in a region known as an intron. And the goal here was to target this intron where the mutation occurs, remove it, and allow for correct splicing of the exons together. And so this is the CRISPR-Cas. You may have heard about CRISPR. It's in the news a lot these days, but this is a particular enzyme that can be used to target the mutation. The mutation is the red X right there. And by using what's known as a guide RNA, brings the CRISPR molecule, the Cas9, to the correct spot, and then it will cut out that mutation, again, allowing for splicing of the gene together, putting the correct exons, and leading to, hopefully, a functional protein. And so the progress so far is that they have identified guide RNAs that can promote correct splicing of the Ush2A gene, and these have shown formation of protein in cell lines. And so here's just quantification of that after looking at several different guide RNAs. They found one that is up to 90% specific for correcting that mutation. And looking at protein, that's shown right here. So this is a single cell where you're looking at the nucleus of the cell shown in blue, and the USH2A protein is shown in red. And this is with the mutation, so you actually don't see very much red protein. In a treated cell, you can see, it might be faint on your screen, but there are little red dots, little red puncta. That's actually the USH2A protein, and it shows that this treatment is working in these cells. And so their preliminary conclusion is that genome editing with this particular form of Cas9 can restore healthy USH2A RNA and protein in the cell lines. And so for this project, their next steps are to further optimize these guide RNAs. They want to test the strategies in USH2A patient-derived cell lines. So this has been done in a generic cell, but if they get patient cells, they can do it in a stem cell line. And now for the third and final project. This one is called Preclinical Development of Patient-Customized ASO Therapies for Usher Syndrome. And you've already heard about ASOs from Jennifer Lentz. It stands for antisense oligonucleotides. These are short stretches of RNA that can be targeted to specific sites in genes. And so this work is being done at Boston Children's Hospital and Harvard Medical School, led by Geleoc and Tim Yu. And I should mention, Tim Yu is one of the only investigators in the world who's led the first clinical trial for a single patient. So this was done a few years ago, and if you want to look him up, I think he's a remarkable individual, and I'm happy that we've been able to recruit him to this field. They're working with postdocs in the lab, Stephanie Moriak and Haohong Huang. So the hypothesis in this case is that the antisense oligonucleotides, or ASOs for short, can be used to target Usher mutations, USH2A in particular, and restore healthy protein expression. And so the first goal is to design ASOs for blocking abnormal splicing for this particular mutation that occurs in exon 6 of the USH2A gene. The second aim is to use zebrafish and patient-derived cell lines to target ASOs to exons 19 and 20. So this is what the USH2A protein structure looks like. This is just a diagram, and you can see the red X's show where these two mutations that are being targeted exist. One of them is in the early part of the protein in exon 6. The next is in exons 19 and 20. And so in this case, using the antisense, they hope to deliver the ASOs and target the red star mutation. In this case, the hope is that they can block what's called cryptic splicing. Again, Jennifer Lentz mentioned that earlier. It occurs in USH1C, but it also occurs at this site in USH2A. And if the ASO can be delivered, you may be able to skip over the mutation and allow for correct splicing of the USH2A protein. So that's one of the aims. The other one is looking at mutations that occur in both exons 19 and 20. And it turns out this is sort of a mutational hotspot within the USH2A gene. There's 45 different mutations that have been identified in both exons 19 and 20, including the one that's highlighted there in red. The idea is that if you can skip both exons, you may be able to avoid all of those mutations and lead to a correct and functional protein. And so progress, this is the way we look at RNA in the lab. So I won't go into the details of this technique, but we're able to look at both the healthy RNA, which is labeled as WT, that stands for wild type. That's exon 6 and 7 as they would appear appropriately. Exon 6, when it carries a mutation, shows a change in the size of the RNA, and so that's visible as that long strip of dark bands. And then after treating with the ASO, you'll see at the lower end, I don't know if you guys can see it, but there's a very faint band. I put a box around it there. So that is removing exon 6 with the mutation and showing just with exon 7. So that was an encouraging result right there. The other one is shown right here, and this is also a busy looking slide. I'm sorry to show you these kind of data, but this is what scientists get excited about. So in the first vertical column, there's one labeled ASO3, and that was shown to skip exon 19. And in the next column, where it's labeled ASO6, that skips exon 20. And the goal was to actually skip both of them together. So they've used a combination of exons 3 and 6 together, and the box that you see in that column shows that it was effective, and actually quite effective in this case. So those were encouraging results. And so the preliminary conclusion for the third project is that ASOs have been identified that can skip exon 6 and 1920. We think that there's further optimization that's needed for exon 6. The next step after showing that this works in cell lines is to test this in the zebrafish model, and then eventually test this in patient-derived stem cells. And so, again, only 6 months into these projects, there's promising results so far. And so I would like to, again, acknowledge the support of the Usher Syndrome Society and the board of directors, as well as the 3 teams of scientists who have been really passionate about leading this research. And I have to say, it's been a real pleasure for me to sort of be in the middle of all of this, to speak with the members of the Scientific Advisory Board, to the board of directors, and to communicate with the scientists and be able to share all of these results with you here today. So thank you for your attention.